This is for cell communication part 2 for AP Bio. So the first signal pathway we're going to be talking about is the G protein pathway. So the first part in the G protein pathway is that we're going to have the ligand. The ligand is going to dock to the receptor. So once the ligand docks the receptor is going to change shape. And the reason why it's going to change shape is so what we call the G protein can attach itself. So once the G protein attaches to the receptor, in the receptor we have the GDP. And once the G protein attaches to the receptor, GDP is going to be transferred into GTP. So once GTP is made, we're going to go down the cell membrane, and when GTP moves down the cell membrane, the adenylyl cyclase is going to take ATP and is going to cycle it through and give us cyclic AMP, or CAMP to be short. So the cyclic AMP, which is these little dots right here, the cyclic AMP is going to go to the protein kinase, which is, so this is going to be inactive. So once the cyclic AMP bonds with the protein kinase, it is going to become active. It's going to become active. So this allows for the message to be passed on to anywhere needed in the cell. The next pathway we're going to be talking about is the tyrosine kinase pathway. Now, the tyrosine kinase pathway can act as a catalyst to rapidly activate relay proteins. So first, we have our two ligands right here. These are ligands. So the ligands, just like before, are going to dock to the receptors. And this is a receptor, and this is a receptor. So once the ligand docks to both receptors, the receptors are going to come together to give us one combined receptor, which is what we call the dimer. And the ligands are still going to be docked just like they were previously. So inside these little circles, there is tyrosine. And what's going to happen is ATP is going to be converted to ADP. And with ADP, we know we have the little phosphate ion. So what's going to happen is going to phosphorylate each of the little tyrosine attached to the dimer. And this is called phosphorylation because the phosphate is being added to the tyrosine. So phosphate is going to be added to each tyrosine. So then this allows for the message to be carried out into the rest of the cell. Next, we're going to be talking about ion channel receptors. So the ion channel receptors, the little orange dots right here, these are ions, and they are just floating outside the cell, outside the cell membrane. So this right here is what we call the gate. And it's called a gate because it's closed and it's holding off these ions from coming into the cell membrane. So how do we get these ions to come inside? So what will happen is a ligand, this green dot here, the ligand, will attach itself to the gate. Now, once the ligand attaches itself to the gate will open. Just as if I go and hold a door open, people will be allowed to come in. So that's what's going to happen. The hydrogen ions are going to come inside the cell and just be spread out just a little bit. So once the ions are inside the cell, it will continue to be like that until our ligand either dissolves or separates. So as soon as that ligand is gone, the gate will close again and no ions will be allowed to come in. So the ions that did make it inside will stay outside. So from the previous example, if I were holding a door and I left the door and let it shut, no one will be able to come inside and they would just be stuck out there because I've shut the door and I've left it. Just as the ligand has left the gate, so therefore the gate will close again. So once these ions get inside the membrane, it's going to start depolarizing the cell. And what it means when when the ions depolarize the cell, that means that it's going to change the cell's charge to equal the ion's charge. Which most of the time, the ion's charges are negative. So if ion charges are negative, then that would make the cell's charge negative. So after this happens, the message can be passed off to anywhere that is needed. So next, we're going to be talking about the intracellular receptors. So in the intracellular receptors, we have outside of the cell membrane is usually hormones and steroids. So when the steroids, they will diffuse into the phospholipid bilayer, and they will come out on the other side into the membrane. And so this right here is our intracellular receptor. So the reason why it's called intracellular receptor is because the receptor is inside the cell. It's inside the phospholipid bilayer. So once the hormones or steroids, either one, come into the cell, it will become an inhibitor, and it will attach itself to the intracellular receptor. So once that happens, it will move over to the nucleus. And I'm just going to draw a dotted circle. The dotted circle is going to represent the nucleic membrane. So once the hormone or steroid, or in this case it's an inhibitor, but once it attaches to the intracellular receptor, it's going to move to the nucleus, go inside the nucleus, and it's going to, I'm going to try my best to draw DNA. It is going to bind with DNA and start making mRNA with the help of transcription factors, which help start the mRNA process, and then it will carry on to make the necessary proteins. So next we're going to be talking about secondary messengers. And the two we're going to be talking about is the CAMP, which is the cyclic AMP, and the CA and the IP3, which is the calmodulin and inositol triphosphate. Now, secondary messengers, these are relay molecules that are within the cell's cytoplasm. and they are ion-based molecules, and they can move around the cell very easily. And the reason why that is important is because the first messenger we have is the ligand. And once the ligand bonds with the receptor, it can't really do anything because it's bonded to the receptor. So these secondary messengers carry out the rest of it and do the rest of the work so the message can be delivered throughout the cell. Now we're going to be talking about the secondary messenger cyclic AMP. Now cyclic AMP is used in the G protein pathway. So here we have our ligand. And the ligand is going to dock or attach to the receptor, which is here. This is the receptor. Okay. So then once the ligand attaches to the receptor is going to change shape. And this will allow for the relay protein. So this is the relay protein, but in this specific case, this is going to be our G protein. So once the G protein is attached to the receptor, is going to give GTP. So now the G protein is going to go down the membrane. And when this happens, this is the effector. And our effector in this specific case is adenylyl cyclase. So when the G protein goes down the membrane, ATP is going to be cycle through our effector, which is the adenylyl cyclase, and that is going to give us our secondary messenger CAMP or cyclic AMP. So once we have our secondary messenger, our cyclic AMP we have a protein kinase that at the moment is inactive. So here we have our little secondary messengers, our cyclic AMP, and these cyclic AMP are going to go to the protein kinase and is going to make it active. So once the cyclic AMP joins with the protein kinase, it's going to make it active and allow for the message to be sent to anywhere needed in the cell. Now we're going to talk about calcium and IP3S secondary messengers. So here we have our ligand, and it is going to come down and it is going to dock to the receptor. And the receptor is then going to change shape. All right, there we go. And the G protein is going to come and attach itself to it. And once the receptor is, or the G protein is attached to the receptor, is going to get GTP. So then GTP is going to go, and it's going to get phosphorylated, and that is going to give us phospholipase C. So we get phospholipase C from GTP being phosphorylated. So then phospholipase C is going to go and it's going to attach to this structure here, and there's the PIP2. So the phospholipase C is going to break down the PIP2 bond and is going to take this and separate it. And so then that is going to give us IP3. And then this little thing right here, the top of the pipe 2, is going to be DAG, and that's another secondary messenger that can be used elsewhere. So then IP3 comes down, and it's going to go to the endoplasmic reticulum. And inside the endoplasmic reticulum is little calcium ions. So these are calcium ions. So once IP3 okay, IP3 attaches to the endoplasmic reticulum. The channel is going to open, and it is going to allow the calcium ions to come out of the endoplasmic reticulum and flow freely. So then the the calcium ions are going to go to what we call the target protein. And the calcium ions, which is our second secondary messenger, is going to attach itself to the protein and allow the message to be carried through.